Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM.
This is TWiM, This Week in Microbiology, episode number 325. I'm Michael Schmidt, and you're listening to the podcast that explores unseen life on Earth. Joining us today from Ann Arbor, Michigan is Dr. Michelle Swanson, where the University of Michigan is 7-5 this year, having beaten the Ohio State University in Columbus this past 30th of November with a score of 13-10. Welcome, Michelle.
And I did not pay Michael to say that, but our basketball team also beat a ranked Wisconsin team the following week. So life is good here. And we also got a lovely seasonal inch of snow this overnight. So winter is upon us.
And you're also going to play number 11, Alabama, on New Year's Eve in the Rillella Quest Bowl. So True. Michigan is having all sorts of great things happen to its sports teams. Also joining us today from Washington University in St. Louis, Go Bears, where Washington University competes in Division III of the NCAA, is Dr. Petra Levin. Welcome, Petra.
It's great to be here. And I have to shout out our women's soccer team who won the Division III national championship. So
absolutely.
Good for them. That's great.
Yeah, they were second last year and they won this year. So very exciting.
Terrific. And I should mention that we are still in our fundraising efforts for Microbe TV. So if you like our work, please consider supporting us financially through microbe.tv/contribute. Today we have two papers for you that I think you're going to find quite interesting that can sort of all be summed up. Blame it on the microbes. And we're going to hear why I say blame it on the microbes in our first snippet from Petra.
Okay, so this snippet is actually about something that people observed about 20 years ago, that there's been an increase of sort of the rise in the increase of methane in the atmosphere has kind of shifted and increased how quickly it's rising in the past 20 years. Partly this is a paper from PNAS entitled Rapid Shift in Methane Carbon Isotopes Suggests Microbial Emissions Drove Record High Atmospheric Methane Growth in 2020 to 2022. It's by Sylvia England Michelle, Xin Lee, John Miller, Peter Tans, J. Reed Clark, Henrik Schaefer, Peter Sperlich, Gordon Brailsford, Shinji Morimoto, Heiko Musin, and Jianghong Yang Li. And then there's a really nice summary of it in the Washington Post, if you have access to that, entitled, Scientists May Have Figured Out Why a Potent Greenhouse Gas is Rising. The Answer is Scary. And that's by Shannon Osaka. So just a little bit, we've talked about methane a bunch of times. It's produced by methanogenic archaea, which are some of the more abundant archaea sort of in what we would call normal Earth temperatures and areas. They're in wetlands. They're in the cow rumen. We had a nice discussion of them in cow rumen and cattle rumen and what people are doing to mitigate methane production by cattle on our October 11th TWiM, which I suggest people might want to go back and listen to, not only to learn about how people are trying to mitigate methane production in the dairy and beef industry, but also to hear about some really cool experiments and an adorable calf named Sushi. But this article is looking more generally at why methane might be increasing.
Could you tell us, Petra, why we should be concerned about methane?
Exactly.
Is it methane, just the smell, or is there more to it?
It's actually, I think, technically odorless. It's the sulfur that you smell. And in natural gas, it's probably the biggest component of natural gas, which is basically surpassing coal, at least in the U.S., for one of the for the fossil fuel used to generate electricity. And that's actually a good thing. It burns much cleaner than coal, and it actually produces 60% less carbon dioxide, which is another greenhouse gas. But methane is a greenhouse gas, and 1 ton of methane is equivalent to the emission of 28 tons of carbon dioxide over a 100-year period, but it does break down faster. So if we could mitigate it, then it will go away, whereas the carbon dioxide doesn't go away very quickly.
And that's the fancy statistic or jargon that they use in the paper. They call that the GWP, or the global warming potential.
Yes, but basically all it means is that it's 28 times more potent than carbon, dioxide, but it does break down faster. And methane is like natural. It comes, a lot of it's anthropomorphic. Some of it is also from wetlands. The methanogens are everywhere. They say everywhere where there is something warm and wet, you're going to have methanogens. And so cow rumen, and also rice paddies, and then wetlands. And so it's a problem. And so this paper, and then the Washington Post article that summarizes it, this paper is really looking at where that methane that's increased in the past 20 years is coming from. And they used a little kind of neat trick, which is there are 2 isotopes of carbon. There are many isotopes of carbon, but the ones that are important for this there's carbon-12, which is sort of the standard carbon on the periodic table. And there's also sort of heavy carbon, which is carbon-13. And it turns out that the ones that produced by microbes is light. It's mostly carbon-12, whereas that from natural gas is carbon-13. And so they can differentiate where the methane in the atmosphere is coming from by looking at these isotopes. And I want to emphasize here with the natural gas, it's not the combustion of natural gas that's producing the methane, because the natural gas is methane mostly. It's the probably leaking, when the idea is that if it's coming from the fossil fuels, it's coming from leaking from the pipelines, or when they make it, or maybe during the combustion process. And so they basically use different techniques to measure the amount of heavy versus light, with the idea the light is from the microbes and the heavy is from the fossil fuels. And they find actually it's not coming from the fossil fuels, which was their sort of original hypothesis, because we're using more fossil fuels, especially natural gas lately in the past 20 years. And it turns out it's coming from the microbes. So the question is, where are these microbes? So agriculture is certainly a contributor, but they speculate that more of it is coming from these wetlands, and potentially some agriculture. And I don't, if those of you who have grown bacteria within a certain temperature range, they do grow better at higher temperature. And the archaea, the methanogens, also do better, produce more methane, and more is released at higher temperature. So it's sort of a vicious cycle where more methane might lead to increases in temperature because of the greenhouse gas, and that will increase the amount of methane produced by mostly, in this case, it's naturally occurring wetlands that are sort of distributed across the earth. But there's a lot, apparently the big hotspots are in the Amazon and also in the Congo.
And it makes perfect sense.
Yeah.
If you think about it, as you raise the temperature of the planet, the microbes are going to grow faster, and methanogenesis is principally a way to get rid of their waste electrons. They literally dump that electron into CO2, and they make methane, which escapes as a gas, which is the ultimate waste product. It leaves the location of the microbe, so they're not sitting in their own waste. So this is, really elegance on behalf of the microbe, and it's through our own fault that we're raising the temperature. But it's a vicious cycle, because the methane then goes up into the atmosphere and the sunlight's energy is retained, adding to the warmth of the planet, which drives faster metabolism, and it's a vicious cycle.
Exactly. And so the methanogens are not the problem per se. I mean, they are the end problem here, but they are just doing their job. And because they're also doing something important, because in the wetlands in particular, also in the cow rumen or any ruminants on Earth, but in the wetlands, they're the ones important for breaking down like cellulose and things from plants and lignins. They take that kind of big molecules that you can see and, plant matter and take it and make it into methane so that you don't have a buildup of all these plant material, for example. And the cows obviously can use the methanogens to sort of digest all the way down to carbohydrates that they can use. But on the other hand here, the warming temperature is just increasing the production of the methane, which is then increasing the amount in the atmosphere. I mean, I think some questions from this are obviously, with any life form, there's a certain temperature zone at which they stop being productive. So I don't know for these methanogens, there might be some temperature change, and it's probably too hot for us to really be able to do the kind of agriculture and things we need to do. But there is some limit on how high the temp can go before they become inefficient at doing it.
But ultimately, they could adapt to higher temperatures.
Right. They might be able to adapt to higher ones, or we'll select the methanogens that can do these reactions at higher temperatures. And obviously, we can't really get rid of the wetlands because, again, they're serving a very important function, and they're also a carbon sink themselves. So it's, they're consuming CO2, a lot of these methanogens. So it's very kind of complicated. And I think in the paper and in the article in the Washington Post, they talk about the algebra of the problem, because you've got something you need. It's a carbon sink, but also producing methane, which increases warming, but the methane breaks down faster than the CO2. And it's really unclear how all these fit together and whether there will be some kind of end stabilization where you've got carbon sequestration kind of matching the production of the methane and the methane breaking down. But I guess on the other hand, I think it's a good sign that it's not coming from the natural gas, because
it's cleaner than coal.
It's much cleaner than coal, and it's more efficient, much cleaner. Most of the coal plants in Missouri have actually switched to natural gas, and so I think on that hand, for their air quality and actually for greenhouse gas production of CO2, which is super stable, it's a good thing. So, there's a lot of different things going in, and it tells you how complicated it is. But I think they were also surprised as to where it was coming from.
Yeah. And as you say, we can't control the wetlands easily, but there are things that we can control. So anybody who's like freaking out and going to lose sleep tonight, let's think about what we can control. Certainly a number of people have shifted away from a meat-based diet in an effort to reduce the methane emissions from our commercial agriculture. Certainly using public transportation, walking and biking instead of using a fossil fuel car, switching to hybrids, EVs.
I think so. And then I think also this discussion that we had on October 11th, the episode on October 11th, about, at least on the agriculture cattle side,
right,
really kind of trying to change what's in the rumen, which is fairly, these are really clever ideas to do it on a way that could be done on a large scale for ag.
Absolutely.
And the simplest thing you can do is lower your thermometer or your thermostat in your home.
Yep.
I mean, you can always put on a sweater, as my mother said.
And warm socks.
And warm socks, yes. Especially you probably need those in Michigan today. I heard from my brothers in Chicago that it's very cold up north.
Yeah, but it's weird because in just a few days, it's going to be in the mid-40s.
Oh.
Which isn't right. Yeah.
No.
What beautiful work, if concerning.
And, what always has fascinated me is that microbes can fractionate isotopes. They can actually select the isotope of the particular element that they want to incorporate. It just so happens that in this particular case, it's the most common carbon-12. But I know in the case of sulfur, they can actually fractionate everything from sulfur 31 all the way up to sulfur 35. And you can actually tell who's there by the isotopic ratio. And so you need a mass spec, and you can figure out who's doing what based on the isotope that's being fractionated by the microbes. So the microbial enzymes are really pretty finicky about the carbon that they are using.
I thought that was fascinating. I also, though, was thinking about fossil fuels, many of which come from not archaea, but from other microbes, plants, and then smaller, other
invertebrates.
And so that is not as big a contributor as the microbes themselves. I thought was also kind of fascinating. So,
well, thank you, Petra. And now we're going to move on to a paper today that is, again, absolutely fascinating. But again, it's going to take the moniker of Blame the Microbes. And Michelle's going to bring that to us. Michelle?
Thank you. The title is A Temporary Cholesterol-Rich Diet and Bacterial Extracellular Matrix Factors Favor Salmonella Species Biofilm Formation in the Cecum. This paper is by Alonso Cruz, Megan Schrieg, and John Gunn from The Ohio State University's Center for Microbial Sciences. And it was published this month in MBio. It's open access, so available to you online. So they focus primarily here on Salmonella typhi biology using Typhimurium as a model. Why is that? Well, right now, there are estimated to be about 9 million cases of typhoid fever each year across the globe. And it's especially a problem in regions where the drinking water and the wastewater are not kept separate. So, for example, in countries like Pakistan, India, Bangladesh, Africa, and Latin America, there's a high burden of disease. And unfortunately, in 2016, an extremely drug-resistant strain of Salmonella typhi emerged, and it is now resistant to all of the common antibiotics. Fortunately, it can still be treated with azithromycin, mixapenem, and imipenem, although recently some strains, these extremely drug-resistant strains that are also now resistant to azithromycin have emerged. So the options are narrowing for treatment of this particular pathogen. There are vaccines available. There's a live attenuated or an inactivated vaccine that's available. And here in the U.S., most of the people who have typhi had traveled to an area where it's endemic. So currently the CDC recommends these vaccines for people who are traveling to those regions. So what is typhoid fever? Unlike Typhimurium, which typically causes a self-limiting gastrointestinal kind of diarrhea, typhoid fever can cause a systemic disease. And that's because, for a couple reasons, but all the Salmonella have a type 3 secretion system, which equips the microbe to inject a range of virulence factors directly into the host cell and alters their biology. So in particular, typhi and Typhimurium can induce their own ingestion by cells that are normally not phagocytic. So when someone swallows food that's contaminated with Salmonella, once it gets into the GI tract, it can actually induce its own uptake by M cells, which are professional traffickers across the epithelium down into the kind of lymphoid tissue under the epithelial layer. And that's rich in macrophages, but again, the bacterium can trigger their own uptake into these cells and then survive in vacuoles and replicate, and therefore gain access to the lymphoid system, spread to deeper tissues. And in particular, they can gain access to the gallbladder. So that is an organ that stores bile, which our liver makes, and the gallbladder releases it into the intestine to aid in digestion of our food. So this can become a kind of a chronic release of Salmonella because John Gunn and others have shown that Salmonella that gains access to the gallbladder can form biofilms, which are these very hardy, resilient, adherent colonies, populations of bacteria on gallstones. And from there, they can occasionally, be shed into the small intestine, and you can set up a long-term asymptomatic super spreader of Salmonella typhimurium. You may have heard of Typhoid Mary. Would you like to tell us, Michael, more about Typhoid Mary?
Sure. poor Mary Malone. She was an immigrant from Ireland, and she came to the United States, and she earned her living by being a domestic cook for the rich and famous in high society in the New York area. And she often got work in the summertime working at their summer homes, specifically in Oyster Bay, New York, where President Roosevelt, not the second one, but the first one, coined the term the summer White House. And she was most famous for her peach ice cream. And the peach ice cream is going to come into play because what's ice cream made out of, folks? Cream. And what is ice cream high in? And that answer is fat. And so you can understand where this might all go. And poor Typhoid Mary was actually imprisoned for a good portion of her life because a very famous epidemiologist figured out and he did good old shoe leather epidemiology, identified the peach ice cream as the culprit. And the consequence is George Soper, which was the famous sanitary engineer, he specialized it, and the state of New York locked her up. And she literally went to Sing Sing. And the oftentimes, this is an ethical question, was Mary Mallon treated justly? And if you think about it in today's world, it makes for a good ethical case because she literally became contaminated. As we're going to learn from Michelle's presentation of the data, we're going to figure out and be able to answer the question whether Mary was treated justly. And I dropped into the show notes a little biography on Typhoid Mary.
I think it is a very interesting question because she would refuse. She refused to switch jobs, and she would try to
change her name.
Change her name. This is what she wanted to do, and even though she was clearly a carrier. So I mean, the ethics go both ways. I think in this, it
does indeed. It does indeed. Yeah. They knew she was contaminated, but there were no antibiotics in 1910.
Yeah. Yep. So for the last 30 years of her life were spent in a quarantine in a small island in the East River of New York. So,
yes.
Yep. Yeah. So why focus on cholesterol here? Well, as I mentioned earlier, John Gunn and his colleagues had demonstrated that Salmonella can form biofilms on gallstones. And I didn't know this, but gallstones are predominantly made of cholesterol. Not only that, but they are a site of long-term persistence for microbes. And that's been shown both in animal models, but also in patients that have chronic colonization. We also know that cholesterol promotes gallstone formation. So there were lots of arrows kind of pointing to let's look at the gallbladder, let's look at cholesterol. So their approach was very systematic. They used a mouse model and they put half of the mice on a 6-week diet that was of cholesterol-rich food and the other half just on their normal diet for 6 weeks. And then all the mice were put back on the normal diet for 2 weeks and then infected with Salmonella typhimurium. So in the mouse model, this does colonize, so it's a good model for this long-term persistence. So they then measured the colony-forming units in feces for up to 21 days of viable bacteria. So we can see that in figure 1C, compared to mice that were on the regular diet, those who got the high cholesterol diet had a really impressive increase, logs of increase, higher burden of Salmonella in their feces. They also went out then and did an organ recital where they isolated the spleen, the liver, the small intestine, the mesenteric lymph node, the large intestine, and the cecum of these mice that were on either diet. And here they were able to find that it was the large intestine and the cecum that showed very little of Salmonella typhimurium in mice that were on the normal diet, but again, a couple logs higher burden of Salmonella if they had been subjected to that 6-week high fat, high cholesterol diet. So clearly the diet was having an effect. And again, the literature had pointed to the gallbladder, so they looked there next. So they did another set of animal experiments, and this time they were using the wild type strain And they also knew of a number of factors that contribute to biofilm formation. So they took advantage of the literature and strains that lacked 4 different components that are known to be important for biofilm formation, and compared them then in their ability to colonize the gallbladder. So in figure 2E, we see that, yes, even as early as 2 weeks after exposure to Salmonella typhimurium bacteria, there is a very impressive increase in colony forming units in the gallbladder. Same is true at 21 days post-infection, but by 36 days post-infection, there were no longer colony forming units in the gallbladder for either the wild type or the extracellular matrix proteins. So that first experiment showed that, yes, extracellular matrix capacity, production capacity, did contribute to early colonization in the gallbladder, but by 36 days, it was not sufficient. So they then went out to 36 days and were measuring now just the shedding of Salmonella in the feces. And here they find that by 21 days, 26, 36 days after exposure to the Salmonella, even the mice that had gotten the mutant that couldn't make the Salmonella that
biofilm,
make extracellular matrix, biofilm, was essentially cleared from the mice. But those who could, the wild type, even 36 days later now, they were still colonizing, despite the fact that the gallbladder was no longer colonized. So
poor Mary. Poor Mary got the bad strain from her ice cream.
And this also points out that the gallbladder, while it's a contributor, is not the only culprit. They've got to look elsewhere to explain what this long-term reservoir is. So they do some very systematic histological studies. And I'll focus, for example, in figure 3B, they do histopathology of the cecum. And this is a little pouch that basically connects the small intestine to the large intestine. And what they find is that even 7 days post-infection, there is considerable inflammation in mice that have been given the wild type typhimurium, but only if they also had that 2-week period, or 6-week period rather, of the high fat, high cholesterol diet.
And again, you don't have to be a pathologist to figure it out. You can tell this, folks, and this again is an open access paper, so you can see these beautiful slides that the authors are offering us. I mean, it's absolutely crystal clear that what's going on here.
Yeah. And they also are rigorous and do quantitation, and that's also shown. So they've got quantitative data as well. So they now show that, yes, you need the cholesterol, but you also need the capacity to produce extracellular matrix in order to see this inflammation. So they next want to look, well, is cholesterol there at the site of the inflammation and the damage? So they're able to actually measure cholesterol levels in the cecum. And again, they can compare different organs. So they look in the serum, the liver, the small intestine, large intestine, and cecum, and they measure cholesterol levels on mice that had the normal diet or those that had the 6 weeks of high cholesterol diet. And again, kind of as the data are beginning to point, it's in the cecum, they do see higher levels of cholesterol that has accumulated on the mice that were fed the high cholesterol diet for 6 weeks. And this is despite the fact that now it's been, 5 weeks since they stopped that high-fed diet, and they're still seeing excess cholesterol in the cecum. So this looks to be a persistent change. So the next question was, well, okay, we're seeing this with typhimurium, which, again, typically causes just not just, but it causes a self-limiting diarrhea. But they want to investigate typhi in this model, because as we mentioned, it's Salmonella typhi that causes this persistent infection that Typhoid Mary and many others have suffered from. So they do similar experiments with infecting the mice now with Salmonella typhi, And this is after the mice have been kept on the normal diet or, again, had that 6-week exposure to the high cholesterol diet before doing the experiments. So again, they do the organ recital, and they see only in the large intestine and the cecum are they seeing an increase in the burden and a persistent increase in burden of Salmonella typhi for those mice specifically that were on the high cholesterol diet, but not the normal diet. So again, they're being very, very careful, very systematic. They do a lot of microscopy to investigate what's happening. And in particular, they were interested in where the bacteria were in the cecum. And for this, I'll highlight figure 6, panel I, where they used immunofluorescence microscopy to look at the position of the bacteria within that organ. And they labeled Salmonella here yellow, and then they used an antibody labeled red to see their attachment organelle, which is called curly in this case. And then they labeled the host epithelium in the cecum with a green fluorophore marking where the villain is. That's a epithelial protein. And what you can see is in the mice that were exposed to the high cholesterol diet, we see a close juxtaposition of the bacteria with the epithelial layer. Whereas the mice that had the normal diet, the bacteria that are there are more in the lumen. So they're not abutting the host tissue. So through these experiments and many others that I didn't describe in detail, they've established that although previously chronic Salmonella carriage was associated with biofilms on gallstones in the gallbladder, here they show that dietary cholesterol, which again contributes to gallstone formation in people, also supports persistent colonization within biofilms in the cecum of mice. And these are mice, it's important to know, whose otherwise their gut microbiota was intact. So this could then be a source for persistent fecal shedding that they see in their animal model. Not only that, but there have been some studies published recently, epidemiological studies in regions where Salmonella typhimurium is endemic. And they have shown that persistent carriage in young patients, but also adults who are shedding Salmonella typhimurium and also Salmonella enteritidis. So although we classically describe that persistent colonization and shedding with Salmonella typhi, they have documented it that there are some strains of Salmonella typhimurium and enteritidis that also can colonize persistently and be shed in the feces. So it will be interesting to understand what is the particular molecular contribution that cholesterol is making in the cecum to facilitate these persistent biofilms. And optimistically, we can now think about new therapeutic strategies to prevent or disrupt this colonization, so we can prevent colonization and chronic infection and dissemination into the water supply. And then in areas where it's endemic, of course, if that water is used for irrigation or for washing vegetables, it can then spread by the fecal oral route and maintain this colonization in the human population.
And it really explains why all of a sudden we have to recall lettuce shipments and spinach shipments and all of these contaminations that we're seeing, because simply people continue to shed if they're on this lithogenic or high cholesterol diet. And oftentimes many of the workers who work in these fields do indeed consume high-fat diets because it's inexpensive, as well as it supplies critical energy so they can work in the fields all day. And, they don't have access to sanitary facilities at the ready, so they're often going in the fields, and, it perpetuates the cycle. I also want to point out that figure 7 in this manuscript is really quite remarkable. It's a nice graphical summary that describes this whole model so you can get an overview as to what's going on, in addition to the beautiful histological and quantitation experiments that these authors offer. And it's one of the advantages of an open source publication.
It's also helpful because you can visualize particular terms that are used in the paper, like what is the cecum? They show it here. They identify a number of those biofilm constituents that were used in some of their beautiful immunofluorescence and histology. So it's a great figure. I agree. Tell us about the author.
The first author, Alonso D. Cruz, grew up in a small town in rural Mexico. And I should point out that he's a research associate. And in spite of limited opportunities as a young child, he embraced biology when he got to university in Mexico City, where he developed his profound interest in infectious diseases, particularly the intricacy of host pathogen interactions, which this paper is truly an elegant example of, including the immunology and the microbiology, which he has clearly demonstrated he loves. He gained real hands-on experience, like many of us, when he pursued advanced studies offering him an opportunity to earn a master's degree. It was here he learned how to formulate hypotheses, plan experiments, and realized that he was truly capable of contributing to the scientific field. He then found his way to the Nationwide Children's Hospital in Columbus, Ohio, where under the mentorship of John Gunn and Megan Schrag, he gained valuable expertise where he was able to offer critical support and realize the value and importance of multidisciplinary collaborations. And I think we've seen that in the beautiful work he has offered to us today. His advice for junior colleagues or aspiring scientists is to stay curious and be persistent. Research can be full of challenges and unexpected results, but it's important to embrace those moments as opportunities to learn and grow. Also, he offers don't be afraid to ask questions or seek advice and mentorship. Science is a collaborative effort, and we all learn from each other. Lastly, while technical skills are important, developing strong communication skills is equally critical for sharing your findings and advancing your careers. And he offered to us in his remarks that he really learned a lot preparing this paper. And he also offers, pay attention, all you admissions directors, as someone preparing to pursue a PhD, I have realized that the ability to convey complex ideas in a clear and engaging way is just as valuable as the research itself. Finally, he would like to advise junior colleagues to find balance outside the lab. Research can sometimes be frustrating and stressful, so it's essential to take your time for yourself and recharge. Personally, Alonso makes sure he plans weekends with friends and enjoys his hobbies that allow him to de-stress and explore new skills unrelated to science. For example, he loves traveling, cooking, painting, biking, and watching anime. And these activities not only help him relax, but also provide him that needed fresh perspective that can inspire the creativity for his day job or his work. Maintaining this healthy work-life balance is crucial for sustaining long-term success and a continued passion for research. He offered that he is looking for his next evolution, if you will, into microbiology and immunology, and is hoping that this publication and work experience he's gained will help an admissions committee in their evaluation of his application. And I think he would make a welcome addition to any school. I mean, he's clearly found his niche, and he is quite good at it. So that was what Alonso offered to us.
And I love the maturity and how thoughtful he is in just thinking about his own path and future. Great advice.
And remember, this individual came from a small rural town in Mexico, where he didn't have access to STEM education and all those other things. He had to wait until he got to university. So many of us today are fortunate in that STEM education is not only offered in middle school and high school, but it's also really tuned up in college. And he was fortunate that he went to University of Mexico City, where he really got a good dose of it.
Yeah, and I'll add that I was a biology major at an Ivy League school, but yet I never did any research as an undergraduate. It just didn't occur to me until spring term of my senior year, and by then it was too late. So my first real lab experience was after I got my bachelor's degree, and I worked as a technician for a few years, and that was really inspiring and launched the career I've had.
Yeah, I think getting into research early is something we really emphasize because it gives students like a hands-on while they're learning the material, and the material is kind of dry, frankly. And so to be able to do the research makes it more exciting. I do want to correct one point that we had about where the contamination is coming. And for ag, I think right now people think it's mostly coming from the irrigation water and possibly contamination from the animals, not from the farm workers.
Oh, yes. I didn't mean to say it was all from there. But
I mean, that's the big source is probably from animals. It's probably mostly Salmonella, not typhi, but Typhoid Mary.
Yeah.
Right.
And other things, E. coli.
And other things.
And worse things than Salmonella. So,
well, thank you very much, Michelle and Petra, for your two papers that blame the poor microbes.
They do good. The microbes give, the microbes take.
Yes, we just have to learn to coexist with them.
Well, so if continue learning so that we can protect ourselves from the good ones and leverage the bad ones and leverage the good ones.
Exactly.
Absolutely.
Yeah.
So show notes will be at microbe.tv/TWiM. If you like our work, please consider supporting us financially. It helps us to continue to make these podcasts free for everyone on the sites that we post to. We'd love to get your questions and comments. You can write to us at twim@microbe.tv. Michelle Swanson is at the University of Michigan. Thank you, Michelle.
My pleasure. Thank you.
Petra Levin is at Washington University. Thank you, Petra.
Thank you.
And I'm Michael Schmidt, and I'm at the Medical University of South Carolina. Please join us next time for the next episode of TWiM. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. See you next time on This Week in Microbiology.